Good afternoon, everybody. Super pleasure for me here today to be, playing Michael Bronstein, which every now and then I get to do. And today is my pleasure to introduce Kacper and Francesco, which are going to give us sort of like a double barrel talk about their new and shiny generative framework for conditional flow matching, which is called metro flow matching. And they're sort of like thinking about using this type of approach for, image recognition tasks, molecular modeling, and all other sorts of kinds of things. So Kacper is a PhD student currently at University of Oxford and working in the interface of machine learning and chemistry and biology under the supervision of Michael Bronstein. Before he was actually at the ETH Zurich. Francesco, on its end, it's working currently in Valence, also in interface of ML, chemistry, and biology. And he was also before, in his previous life, at Oxford with Michael and Pietro, and did his PhD in math at UCL. So, well, I won't go too much more into the details, and I'll pass the ball to your side, and we look forward to hear about these new advances.
Perfect. Thank you very much for the introduction, and let me show the slides. Okay. So hello, everyone. Again, I'm Kacper, a PhD student at University of Oxford. And today, alongside with Francesco, we will be presenting our work on metric flow matching for smooth interpolations on data manifold. And yeah, we're excited to share our findings with you today. So let's dive in. So let me begin by explaining the primary problem that we try to tackle in the research. And this is all about understanding the system dynamics from sparse noisy measurements. And a main problem in many biomedical applications is the lack of access to complete time trajectories. And we often only have those cross-sectional sparse samples. And as an example, we focus on the primary example of these conditions when we have cross-sectional samples, which is the single-cell RNA sequencing and reconstructing the trajectories of single-cell dynamics. So what is single-cell sequencing? Single-cell RNA sequencing measures gene expressions at individual cell levels. So however, as I explained before, there's a problem here. The process is costly and destructive. And this means that each cell can only be measured once before it's destroyed. So this results in exactly the data that is sparse, unpaired, and cross-sectional. So our aim here is to reconstruct the system temporal dynamics from such data. And this is widely known as a trajectory inference problem. So as an example, on the slide, you can see the low dimensional embedding of single cell data. It is measured along different time spans, and like each color represents different time it was measured at. And what is exactly important here is that those pairs are unpaired, so we don't know exactly what is the correspondence between the cells measured. However, to further motivate the work and the framework we introduced, there are also closely related problems parallel to trajectory inference. So to think about the settings that our framework is useful, it is when we have source and target distributions that are both informative priors, and we care about interpolations between them. And this is the case for both LiDAR navigation and unpaired image translation problems. So first, see the landscape model on the left. We have one source distributions marked as red points, and then we also have the target distributions marked as blue points. And now we would like to find a map for which interpolants stay on the surface on the manifold that is defined by the data. And similarly, on the right, we have the unpaired image translation problem. And we want to convert images from one class into the other, like dogs into cats, but also keep semantic features of source images. And the question here would be how the transition path should look like to keep the most semantic features from the source. So as a background, let's now discuss the current state-of-the-art solution for trajectory inference problem and what we actually building, extending our framework on, and this is conditional flow matching. So formally, it is a framework that finds a push-forward map from source to target distribution, and most importantly, does that without the need for simulation during training. So essentially, conditional flow matching generates this map using a time-dependent vector field. And this is in line with continuous normalizing flows. And for learning, conditional flow matching only considers paired samples from source and target distributions and paths between them. Coupling could be either drawn at random or sampled with some pan like optimal transport. So once we establish these data pairs, conditional flow matching employs straight line interpolants. So you can see in the equation that xt it's ensured that like at time equals 0, we're gonna have retrieve x0, at time equal 1, we're gonna retrieve x1, and everything else is just a linear interpolation. So having that, let's move on to the conditional flow matching objective. And so now having the straight line interpolants, how do we get the vector field? So we parameterize our vector field as time and x-dependent neural network. And the objective is then simply regressing the conditional vector field in expectation. And the traditional applications of such framework is generating new samples from some easy to sample and informative prior. For instance, on the picture we have we're generating half moons from 8 Gaussians, but we could also generate images from noise. However, in our setting, the metric flow matching, we mainly consider a different case where the scenario is that, again, the both source and target distributions are informative empirical distributions. And here we not only care about generating the target, but also about meaningful interpolations. And now Francesco will outline why conditional flow matching might be limited in that regards. Let me share stuff.
Okay, I think you can hear me, right? Yeah, so I will now quickly give you an overview over the next 10 minutes of why we have that term in the title of our paper, which is metric. So essentially, what's the where the geometry enters the picture here. So as Kacper mentioned, classically, conditional flow matching relies on straight paths, which is a way of saying Euclidean geometry. Now, the reason why this works in certain applications like image generation from noise is because typically the heuristics is that if you have a linear-ish type of dynamics, you can do inference with bigger steps. The problem is in applications like specifically in the scientific domains, and for example, for trajectory inference, the dynamics that you want to actually reconstruct more often than not is nonlinear, right? So straight paths will not be able to capture the underlying trajectories, which concretely means, as in the picture here, they will stray away from the manifold where the data lives and end up assigning non-zero probability to regions of space that should not be traversed. So this is a sort of a way of saying that something that people have adopted in many different domains, which is the manifold hypothesis, right? So even though the data we observe live in a linear high-dimensional space, they're actually the degrees of freedom we care about are in fact, they in fact belong to a lower dimensional curved manifold M. And this is a property, for example, satisfied by cells when embedded in the space of gene expressions, mostly because the degrees of freedom that we usually are interested in are much lower than the high dimensional coordinate representation of cells. So we know that in many applications, the manifold that is actually where the data lives is a curved lower dimensional space. And ideally, we would like the interpolants to be supported on such manifold, so they might give rise to like more meaningful matching. The problem is that operating on a manifold is typically challenging. So you need different charts, different coordinate systems, and people have even observed that when you define a generative modeling on a lower dimensional space, this may incur overfitting. So we would like to avoid having this extra overhead of characterizing what a manifold actually is. And we are, to circumvent such an issue, we adopt a perspective that is typically called metric learning, where you don't change the ambient space, but you change the geometry of the ambient space. So in other words, we don't really need to know where the manifold is. We just use a common principle, which is the samples we have observed are representative of where the manifold is going to be in our space. In other words, the interpolants that we design should remain close to the data points. If we can achieve that, then the interpolants will approximately stay close to the manifold, even though we haven't defined the manifold explicitly. And this, in a nutshell, sort of the principle behind metric flow matching, which is a generalization of conditional flow matching, where essentially you learn probability paths to stay supported on the data manifold. And we can do that without incurring simulation. So it's relatively similar in terms of computational cost. So how do we achieve that? We need to sort of change the geometry of the ambient space, which formally means we need to define a new metric in the ambient space, right? So first of all, in our work, what a metric is, essentially this is a function that assigns to any vector x in your Euclidean space a symmetric positive definite matrix G. So this matrix essentially is a norm, and this norm will change based on where in space you are evaluating such a norm. The intuition here is the norm should be higher as you move away from the data, because when that's the case, you can then use a sort of kinetic energy or this type of quantities to regularize the flow and penalize the velocity the more when it's away from the data. So a data-dependent metric is essentially an assignment that gives you, for any point x, this positive definite matrix that will depend on where the samples in space are. And for simplicity, throughout our work, we actually restrict to diagonal matrices. So concretely, you just assign a vector with positive entries to each x. Right, so we have a metric now in our ambient space. And what we actually prove in our paper is that under very mild assumptions, you can always construct a metric whose shortest paths, also known as geodesics, stay close to the samples you observe. So just to give you an intuition, imagine that the data you observe are like in the picture, then you can always construct a metric whose, for example, trace, so the sum of the eigenvalues is much, much larger as you move away from the data, and is lower when you move close to the data. And Kacper later is going to present you such a metric. And if that's the case, then it turns out that, if you start from example from the left and you want to go to the right, the shortest path, also known as geodesic, according to such a metric, will not be a straight line, but it will bend and remain close to the data points because this is where the cost is lower, okay? So in other words, the metric indicates where in space your interpolants should go to minimize a certain objective, right? Okay, so all good. In theory, if we have access to geodesics, we are done. This could be our interpolants, and then we can feed our interpolants into the flow matching objective. The problem is, essentially, for almost any non-trivial metric, there is no analytic expression for geodesics. And the only way of deriving geodesics is through costly simulations. So to avoid simulations, instead of coming up with the exact geodesics, we try to approximate it in a learnable way. So what we propose is to define interpolants that now they no longer just depend on time, t, but they also depend on additional parameters, eta. So the way these interpolants are built, you basically take the straight path you saw before, and then you add a nonlinear correction. Now, this nonlinear correction is made of two terms. The first one, which is only time dependent, is key because it ensures that this nonlinear correction vanishes at times 0 and 1. And because of that, you're always guaranteed to satisfy the boundary condition. So you will always retrieve x0 at time 0 and x1 at time 1. The nonlinear term is instead, for example, an MLP that depends on x0 and x1 and will add a correction when you move away from the boundary. So it can allow you to sort of bend the path according to some objective. So all it's left to do is defining the objective. Once we have this objective, we can train a model and find the optimal additional parameters eta. And this is where we, if you like, rely on Riemannian geometry again, because it turns out that if you give me 2 points x0 and x1, and you give me a metric G, then the shortest path between x0 and x1 according to G, again, what is known as a geodesic, will be the global minimum of this convex Dirichlet energy functional. Now, as I mentioned earlier, finding such a global minimum is challenging. It often requires simulations. So what we end up doing is finding the minimum of this Dirichlet energy over the class of interpolants we designed above. So our basically additional loss now samples x0 and x1 according to our coupling, whether it's independent coupling, optimal transport, doesn't matter, whatever we end up choosing, and then estimates this Dirichlet energy over the class of interpolants. And this way, we end up learning paths that sort of behave like geodesics because they avoid regions of space where this metric is large, which are exactly those regions of space that are away from the data samples. Something that should be worth emphasizing here is that the objective is in fact a regularization. As you can see, it acts directly on the velocity of the path, and it penalizes more the velocity the more you stray away from the data samples. So this is in a nutshell the framework, and now I will leave it to Kacper to actually describe how this works through algorithms, the choice of the metrics, and the applications.
Okay. Okay, so let me go now first through the algorithms. And our framework consists of 2 steps. The first one is going to be finding the interpolants and training the network phi. And the other one is going to be regressing and actually finding the vector field. So again, we have this non-linear correction factor, which is parameterized by the network phi. And as an input during the first step of interpolation, we take our source target distributions data-dependent metric. Then we get our pairs of samples from source and target distributions. We either sample them at random, but usually they are sampled actually with optimal transport. We also uniformly sample time for each pair of the x0 and x1 samples. And having that samples, we have our traditional like linear interpolant from conditional flow matching marked as green, which basically results in a straight line in the ambient space. Now we add to this our non-linear correction vector marked as red. And again, we have the network phi, but we also guarantee ending up at x0 and x1 due to this factor t times 1 minus t. And this results in our like initially assigned interpolant marked in purple. Having that, we can calculate the corresponding velocity vector in the ambient space when the derivative of the network can be found, of the network phi can be found with autograd, and the rest is simple derivative. And finally, we use that to calculate the kinetic energy with respect to data-dependent metric. With this optimization, the network is trained so that the points along the interpolants lie as close as possible to data points. And in practice, as Francesco mentions, we act penalizing velocity sort of also acts as regularization. Because if, for instance, we would use the norm or like the trace of the metric, it could potentially result in overfitting. So after this converges, we finish this first step of the training. So again, we learned interpolants by learning the network phi. And now we will try to regress against the vector field in the second stage of our training. Okay, so here I'm showcasing the second stage of our framework again. And this is essentially traditional flow matching, but with added nonlinear components to interpolants. So we've highlighted in red the modifications that need to be made to adapt it to metric flow matching. So firstly, we have our pre-trained frozen network phi that is added to the interpolation. And then we calculate the derivative with respect to that and plug it into the flow matching loss. Now it's to embrace the Riemannian formalism. It's also like with respect to data-dependent metric. So yes, that would be it for the training itself and for the training procedure. Once it obviously converges. So just to give slightly clearer picture of our framework, we wanted to compare it with like similar extension of flow matching, namely Riemannian flow matching. So just to summarize, Riemannian flow matching aims to extend the flow matching to train the normalizing flows on manifolds. However, there are like two main significant differences, and I believe from that also this results in like different potential applications. So first, our approach relies on metric directly induced by the data. And in contrast, the Riemannian flow matching relies on metric that is provided by the user of the framework. It has to be given, and as usually independent of the data. And the second very significant difference is that we've developed metric flow matching so that it doesn't require any simulation and doesn't require providing a closed form geodesics. And this results in complexity that is very similar to just conditional flow matching. On top of regressing against the vector field, we training first the interpolants, but because algorithms are super similar, it usually converges even faster than conditional flow matching part. So at top twice the complexity. So in contrast, the complexity of Riemannian flow matching stays the same only if the closed form solutions of geodesics are given. Otherwise, if we have manifold without closed form geodesics, the algorithm requires costly simulations. Yeah, and again, just to stress, I believe that just from both of these points also follows different applicability. So there's still one detail left of our design that I would like to cover. And this is exactly how in practice we could construct a suitable metric. And the main property we would like from the metric is that either the norm or the trace of the metric should be smaller closer to the data and larger away from the data. And this can be achieved, for instance, by the metric introduced in a paper called locally adaptive normal distribution, which is LAN for short. And this is why we also call our metric LAN, and we follow basically the approach of the paper. So The land is constructed by calculating the MSE between our point and all the points in the data set across dimensions separately, and then we're doing the weighted sum, and the sum is weighted proportionally to how close, according to radial basis function, our point of interest is with regards to points in the data set. And then it's obviously all summed up, and we take the metric to be inverse of that with some regularization parameter epsilon. And the plot here I'm demonstrating is indeed confirms that our metric works as intended. Points lying outside of the manifold support and outside of the data support have a significantly larger trace than the one lying near data points. However, land is not without a few issues. For instance, the search for hyperparameter sigma in RBF kernel, especially in higher dimensions, proved difficult. Land is also gets more computationally demanding, especially as it's cased linearly, like with number of points in the data set and with number of dimensions. So because of this, we also explored another approach detailed in the paper called geometrically enriched latent spaces by the same author as land. And this method simplifies things by clustering the data first. And then we use these cluster centers as reference points. And we applied the weights. We're also doing the weighted sum, but this time the weights are learned by the radial basis function network. And the goal of the network during learning, how do we find the weights, is by basically minimizing the metric entries close to the data support of the manifold. And in practice, we use land in lower dimensions and radial basis function metric in higher dimensions. So I believe with that, we've covered all the core components of metric flow matching. And the first one is the metric design. Then we learn the interpolants, and then we train the vector field. Having that, we can now transition to presenting the experimental results. Okay, let's dive into experimental results. We first revisited the ARCH experiment, which we've discussed along with the algorithm, I believe, and presented them on the slides. So we use the target and the source distributions directly as the data support in our metric. In contrast to that, and this is also different applicability, we have the LiDAR navigation problem, where the metric itself is actually not like, we have different set that is the data support of the metric. Once we have both source and target distributions that are like 5 Gaussian projected on the different sides of the manifold. And yeah, in both cases, it clearly, we can see that metric flow matching adheres to the underlying manifold defined by the data points, and it leads to more meaningful matchings. And obviously, because conditional flow matching does not consider that problem, it doesn't even consider that at all. So following, we have the unpaired image translation problem. Here we generate cats from dogs. And we can see that like for both quality and quantitative comparison, metric flow matching retains more meaningful semantic input features. And this is achieved through smoother interpolations on the data manifold. Following, we move on to our main problem, which is the trajectory inference of single cell data. So we have access in our data set to either 4 or 5 time points. So to test our performance, we leave one time out. And then we compare how well the interpolated left time point compares to ground truth data. Having that, also we tested in both low and high dimensional settings. Usually it's a common practice to actually get in bio community to get this gene expressions and project it into using PCA to 100 dimensions. And we have clearly outperformed like conditional flow matching marked as red and previous best performing models in both low dimensional and high dimensional scenarios. So on top of the numerical results, we also wanted to provide some intuition why our model works so well in single cell data scenario. So on the slide, you can see that we have the single cell embedding in 2 dimensions. And for conditional flow matching, clearly ignoring the data support leads to trajectories that go through regions that are empty of data, and naturally this is incorrect, while our method avoids this region entirely. And I believe that would be all regarding our results. So now we can move on to like final messages of our presentation. So just to sum up, metric flow matching is simulation free. It generalizes conditional flow matching, and metric flow matching probability paths are supported directly on data manifold. To achieve that, we minimize the geodesic cost formulated as the kinetic energy on data-dependent Riemannian manifold. And again, for reconstructing single cell dynamics, we outperform all methods relying on Euclidean geometry with simple task-agnostic metric. And I believe that would be all from us. And now we could proceed to questions. Thank you.
Well, cool. Thanks a lot. Maybe I'm going to get started with a few points. So maybe just so that we understand a little bit better. In the end here, whenever you're sort of like following the distribution of the data, do you gain or lose in exploration capabilities, right? So let's say that I want to generate a sample point that it's new, right? But in the end, so like when you're doing all these trajectories, you're being sort of like very close to the data that you have present. Do you in general see this as sort of like something that can become more exploratory or less exploratory?
So maybe I think so from experimental results, at least the ones we have, the only one that like directly addressing this question could be like FID that compares the distributions of generated images, and like comparing to conditional flow matching, we managed to like our model actually achieved better score in FID, being that like the final image better reflects the overall distribution of the target distribution.
Yeah, I thought that this was a very interesting result, right? Exactly because of what you're saying, that just by forcing a different path of generation, you actually would end up with better results. Not necessarily about the intermediate points, okay, nobody really cares so much about those, but you actually sort of like converged in something that it's more accurate, right? Or at least sort of like quantitatively. So I think that's interesting too, right? Maybe the advantage is not necessarily in intermediate points of the trajectory, but rather in the final points, which just become better.
Yeah.
Yeah, if I can also add 2 points about that. So maybe something that has to be mentioned here in the context of unpaired image translation is that we operate in latent space, which is often the case here. And we probably, it's not surprising that accounting for data geometry in the latent space is ultimately beneficial, because like we kind of know that when we encode these objects, the encoding is highly nonlinear. So the distances will be distorted. So having Euclidean distances in latent space might not be reflecting of Euclidean distance in the ambient space we started from. So that's an important point. But the second one is about exploration. You're absolutely right in the sense, like, what probably should be one of the things that may come out of our presentation is that if you may be, at least that's also what we are kind of thinking about, is like you may be thinking of objectives that might favor exploration, if that's one specific target of yours. And of course, this is yet again a bias or objective you may have, and work on that rather than just having straight paths between source and target.
Now, of course, the other point of what you're saying is like, well, you know, even if you have something that it's more exploratory, but if it's just generating things that are not realistic, it's also not super useful, right? So in a way, staying within the data distributions, even if it's less exploratory, but it might still be able to get better results, right? You know, ultimately.
Yeah, I think it's a trade-off, probably, like, you know, the way I'm saying, and that's just an intuition, but like oftentimes with straight paths and images, the reason why they might work is that if we have a lot of samples, straight paths are okay, because they can like kind of rectify the distribution, and essentially they're saying infinitesimally any differential equation can be moved linearly. And if you can't see all the distribution, that's all right. The problem are more about when you don't have enough samples, or you can't see some modes, and therefore some form of inductive biases there could help. But of course, in our paper, we did something very simple, which is the metric doesn't depend on the task, doesn't depend on the data type. It was just almost like visual, like validating that even something simple like stay close to the available samples was helpful for a certain task. But of course, you can do much more complex objectives. You can do that.
And you guys don't really care about which data type you're handling, right? You know, because I guess clearly here, so like you can do the stuff with images, you can do the stuff with single cell expression data. So in a way, the data source for you guys is not super important, or is there anything special about the way that you can encode each data point?
No, really. I don't think so. I think exactly this is like more kind of general approach that like we have this task agnostic metric that can be exactly like incorporate any sort of data.
Well, particularly, you know, one of the other points to bring up is this conversation of the single cell data, which also happens in, you know, a lot of many other data types when you basically use sort of like this PCA to cluster the stuff, right? You know, and of course this is a representation, a low dimensional representation of like high dimensional data. So it always brings with itself some type of uncertainty, right? You know, so clearly here, you know, the cells are like, you know, colored orange for days 6 and 9, but not all the cells are the same, right? You know, because they're not clustering all the same, which is, again, you know, that's not the point, because you're coloring them by time. So maybe that's okay even. But, you know, it is always this. So will you always need some kind of low dimensional representation of the data to try to extrapolate these geodesic paths, or you don't really care about that?
No, we don't care about that. This is like simply for visualization. But like as we showcase here, we use the 100 dimensions and even more in the case of latent space, which I believe is 32 times 32 times 4. Yeah.
Well, cool then. So I think if there's no other questions, we'll offer one other possibility to ask a question in the next 5 seconds. If not, then we'll probably close it here until the next time. And we thank our speakers for today's talk and for the beautiful work that they've been doing in this domain. And thank you all for coming.